right, well, hello. Today we're going to take a look at some of the rules of population genetics. We've been studying all about evolution. We've been taking a look at how evolution is related to change in a species over time and how natural selection works. Now we're going to take a look at it from a little bit of a different perspective. We're going to link together the ideas of evolution and specifically to the ideas of genetics, and specifically to how frequencies of genes or alleles change in a population. So that's what we're going to take a look at in today's topic video here. So first of all, what's going on? Just kind of to recap. It's true that Mendel and Darwin never met one another. In fact, they didn't really quite live at the same time. But if they had, they would have had a lot to talk about because their two works really go hand in hand with one another. Genetics certainly explains a lot of what's going on with natural selection, and natural selection explains a lot of what's going on with genetics, for that matter. And one of the things that's important to remember here is that natural selection works on phenotypes. It works on physical appearances. For like our little mice in the picture over here, natural selection is going to pick either white mice or gray mice. It's not looking at whether a mouse is homozygous or heterozygous or anything else along those lines. It's going to say, oh, you're white. Delicious. I can see you. However, underneath those phenotypes, there is a gene pool. There are always genotypes that explain those phenotypes. So yeah, if a white mouse gets picked up out of the population and eaten, that's a couple of recessive G alleles that are getting taken out of that whole population of mice. So even though natural selection works on individuals, we can step back and take a look at an entire population and say, okay, over time, what's going on with this population? Are they becoming more gray? Are they becoming more white? Is the dominant gene becoming more common or less common, etc.? And this is the idea that evolution is actually a study in changing gene frequencies. So we're not just looking at one individual, although one individual either lives or dies. We're looking at the whole group, whole population of mice that can breed with one another and take a look at what's going on. That whole population of mice is called the gene pool, right? Each mouse contains two different alleles, and we can take a look at how common or if gene frequencies are changing. So again, if you take a look at that white mouse little picture there, poor white mice, we could say that, hey, the number of white genes in this population is definitely decreasing as those white mice gets picked off. It doesn't mean they're gone, because remember, these gray mice could be heterozygous. There could still be that recessive gene hidden in there, but definitely it's not as popular as it once was. So let's take a look at how this gets studied. So when we're studying allele frequencies, and that's one way of interpreting how evolution is, we have to take a look at not just the number of white plants, or in this case, white flowers and pink flowers. We actually have to take a look at the number of alleles that are present in the population. So if you take a look at this, down at the bottom, it looks like a bunch of crazy math, but don't worry about it too much. Basically, what it means is we're going to count up the number of dominant genes and the number of recessive genes that are present in the whole population in the gene pool. We're going to use the letter P to represent the frequency of that dominant allele, and we're going to use the letter Q to represent the frequency of the recessive allele. Frequencies are a lot like percentages. They're just written as a decimal place, as opposed to like 100% or 50%, we write it as 0.5. That's all it is. So if you take a look at this first one, and now is probably a good time to hit pause. If you take a look at this first one right here, if we count up the total number of plants, there's 10 plants there, so there's 20 alleles, and if we count them up, there's 14 big A's and 6 little a's, so we can figure out how we do our populations over time. That we would have 14 out of 20 would be the dominant allele, so we get a frequency of 0.7, and 6 out of 20 of them are the recessive allele, so we would get a frequency of 0.3. That doesn't mean that 30% look recessive and 30% look dominant. That means of all the genes in that population, that's the frequencies. So again, you can take a look at the second and third generation here, and you can see that there's something that's going on in the system where in the second generation, that Q allele is becoming more common, and in the last generation, that Q allele now is basically gone. It's kind of a different sort of circumstance. So this is something else that's going on over time, that something is changing. Keep in mind that whenever we have two alleles, that P plus Q, the frequency of P plus the frequency of Q, always has to add up to 1, because frequencies are, like I said, like percents. So P plus Q equals 1. That's a rule that we're going to use here as we move on. So these two guys up in the corner here, Hardy and Weinberg, they were some of the first ones to really kind of put this number stuff down. So you can hate them, not me. But they were the guys that defined what evolution was in terms of gene pools, in terms of allele frequencies. So they said that if a population is not evolving, then over time, even though individuals may come and go, that for the most part, the values for P and Q are going to stay the same over time. That if it starts as, P is 0.7, that if we look generations down the road, P is still going to be about 0.7, give or take take a little bit of, chance error. And that's called genetic equilibrium, or Hardy-Weinberg equilibrium is the other thing that it's called. If populations are evolving, however, we know that those frequencies are going to change a little bit, like in that white mouse example or the flower example, that the dominant or the recessive allele might get more common or less common as a particular plant or animal is being selected for or selected against, as the case may be. So we can do some simple math to actually figure out what frequencies of genotypes are and whether things are changing over time. So if you take a look at this equation over here, this is actually a mathematical representation of a Punnett square, which I know sounds kind of funny. But if you have PP or big A, we would call that frequency P squared. If you have 2PQ, that's the frequency overall of our recessive ones. And if we have Q squared, that's recessive of our these are the frequency of our heterozygous ones, which look dominant. So we could add all of these guys together, and that's our total heterozygous, or dominant looking ones, and the frequency of these guys is our total number of homozygous looking ones. Trust me, I've got some math problems that you can use to practice on. So for right now, take my word for this. Let's take a look. So here's an example. This is a pretty advanced topic, so this isn't going to really show up on your test too much, so don't freak out a whole lot. I'll let you hit pause and take a look at this. Remember I said that equation was basically a mathematic version of a Punnett square? Well, there's your Punnett square right there. And if you take the frequency of one particular allele, that's going to be like the letter P for your dominant one. The frequency of your dominant particular homozygous dominant genes is going to be P squared. You can actually do the math on that. So you can take a look down at the bottom. I have an example problem down here. So from this one that we've got right here, we've got P is 0.7 and Q is 0.3. We looked at this earlier. So that means the number of homozygous dominant ones we have is P squared, 0.49 or 49%. If you take a look at it, it's basically half, or we'd predict it to be half. If you're talking about heterozygous ones, that'd be 2PQ, which is 2 times 0.7 times 0.3, which is actually 0.42 or 42%. And the number of recessive ones would be about 9% Q squared. Total, again, is 100%. And you can take a look at the problem that's up there, and it's usually plus or minus an individual or two, but that's usually pretty close to what we'd expect. So, again, this is a pretty advanced topic, so don't freak out if this doesn't work for you right now. I want you to be exposed to it, come to class, and I'll show you some sample problems and we can work on it. Okay, so Hardy-Weinberg defined genetic equilibrium, a population that's not evolving, by looking at those gene frequencies, and they should be staying the same over time. In order for equilibrium to really happen in the real world, which pretty much never happens in the real world, but for it to happen, there has to be 5 conditions. Number 1, you have to have a really large population. Large populations keep that randomness kind of at check, and make sure that things stay pretty common. Random mating has to be a rule of this. In other words, any female could mate with any possible male in that population. And again, that doesn't really hold in most real populations. Furthermore, you have to have no mutation, or your mutation rates have to be so low that we could pretty much ignore them. You have to be able to say that no immigration or no emigration. Creatures cannot be leaving or entering that population. And then last but not least, there has to be no natural selection. These are the things that has to be happening for a population to stay stable at equilibrium over time. And if you look at that list of 5 things, that's really unlikely. Pretty much every one of those things get violated in some population over time. So they were pretty much saying, well, hey, in the perfect world, for something to not happen, this has got to be held in place. And now we're going to take a look at really the ways that these things can change, because Hardy-Weinberg equilibrium never occurs in a real population. It's always shifting for some reason or another. So let's take a look at closer examples of why these things happen in a population. So, first of all, natural selection, we've talked about this before. One of the biggest and strongest, fastest driving forces of changing gene frequencies is that there's a selective advantage to one or the other phenotypes. And because genotypes are underneath there, if one phenotype is an advantage, then the genes that go along with that phenotype are going to become probably more common over time. So the peppered moth example is a good one, and you can go look this one up in terms of long term. When those pepper moths live in a nice clean environment, the white phenotype is definitely an advantage, but when they live in a polluted, darker environment, then the darker phenotype is an advantage. But keep in mind that what's an advantage under one circumstance or one population can change for another and can change over time, just like in the peppered moss. So that idea of a selective advantage is a selective advantage right now for this particular population. So that's one reason why they can change. Second reason why populations can change is actually just due to chance. This is called genetic drift, and that word drift should give you that idea, that sort of chancy feel, that things can happen random events can go on, like these poor green bugs getting stepped on, or these poor frogs up at the top getting death, I don't know why, but for whatever reason, that a population can get changed simply because a random number of creatures die for no apparent reason that's not related to that particular trait. And we would call this, again, genetic drift. So up in this top group, these kind of darker red frogs all died, again, for whatever reason, we don't know. Maybe they got a disease. Maybe there was a flood. There could be all kinds of things that happened. And all of these lighter colored green frogs are the survivors. Not necessarily because red was a disadvantage, but just because that's what happened in this case. Same thing down here with the dude stepping on the green beetles. It's not that the green beetles was really a disadvantage. It wasn't their size that was a disadvantage. You can see there's bigger ones and little ones. It just happened to be that when the dude put his foot down, clumsy guy, he happened to take out, 2/3 of the green beetles. And again, that's genetic drift. It's not due to one thing really being an advantage, just due to chance events causing a shift for whatever reason in the traits that are being left behind. In terms of population size, and this is that rule that they had Hardy-Weinberg about being a big population, genetic drift is more likely to hit hard a small population than it is to hit a big population. And this is kind of a key idea. And you can take a look at these three graphs that are on here. That top graph is looking at sort of genetic drift in a really small population. That's at n equals 20. And you can see certain things become really common and certain things kind of go away. If you take a look at the bottom one, that population at 2,000, same kind of thing happens. This is what happens with averages. We see this at our school, right? We've got a class of, 10 or 20 people. If we have one or two really bright people move in or move out, it can really change our test scores just by chance. In a class of 2,000 freshmen or sophomores or whatever, one or two smart people doesn't really make a big difference in the overall average. And that's the idea of population size. Smaller populations are more hit by random events than big populations are. Okay, a couple other sort of chancy things that can happen. Something called the bottleneck effect. It kind of goes along with some of those examples I used for genetic drift. A bottleneck is exactly that. It's a narrowing of the population. Again, due to some sort of chance event, only a few individuals survive that bottleneck. There's a huge reduction in population. And so your lone survivors, like in this population up here, happen to all be, it looks like blue and yellow to me. And so the little blue and yellow ones reproduce to make the next generation, and now your new generation is all blue and yellow. Again, it's due to something other than natural selection. It's due to a chance event where your population is really reduced. That's what makes it a bottleneck effect and not just genetic drift. You've got to take most of your population out and only have a few of your survivors reproduce. That makes a bottleneck. Kind of similar to that is this idea of a founder effect, where if you've got a bigger population and maybe they get blown onto an island or get into different sort of ecosystems where they're separated from one another, just by chance, you may have only a couple of individuals founding a new population. So like, for example, in this one, there's not like all these particular creatures are dying here. It's just when a new population gets made, maybe only these brown ones move forward, or maybe it's 50-50, or maybe only the blue ones move forward. The Galapagos finches, Darwin's famous example, is an example of a founder effect. The reason why that particular kind of bird was on those islands is because only that kind of bird got there. It wasn't that a lot of birds got there. The founding population of birds was simply finches, and so finches evolved to fill this whole population of creatures. Another example of bottleneck and founder effect would be what happened with the Ashkenazi Jews. Ashkenazi Jews is a group of Eastern European Jews that, due to historical events, was greatly reduced. So current populations of Jews tend to be related to a very small number of ancestors. And again, so we have bottleneck and founder kind of going on at the same time with those folks. And Einstein, by the way, is a good example of an Ashkenazi Jew. So there you go, your big ideas in terms of population genetics. That a gene pool is all of the alleles in a population, not individuals, but when we pool them all up, how many are big A's and how many are little A's. When we take a look at gene frequencies then over time and how many are big A and how many are little a, we can determine whether or not a population is evolving or whether it's not evolving. And again, this is a definition of evolution in terms of genetics. Hardy-Weinberg had two simple little equations that they used. Remember, P and Q relate to the dominant allele and the recessive allele. So, I'll let you take a look at those two equations again. Don't stress about it too much. But that idea of Hardy-Weinberg equilibrium, for a population to not be evolving, we had to have some kind of not realistic expectations like really large populations. That could be realistic. But random mating, definitely not. The lack of gene flow, probably not. Gene flow means things coming in and leaving that population. No mutation, we know mutation happens. And no selection, and we definitely know selection occurs. So for a population to be under equilibrium, it's unlikely. And then changes in gene pools can be due to things like natural selection. That's what we've been talking about this whole unit. But I want you to be aware that there's some non-selection events that can cause changes in gene pools, cause evolution, not due to natural selection. Things like genetic drift, bottleneck effect, founder effects, things along those lines. So again, if you're looking for a 4, looking for the high level, come see me and we can talk about Hardy-Weinberg in a little bit more in depth. Once the light bulb goes off for you, you'll see it's really not that hard a principle, but it's just kind of a different way of looking at what evolution is. So there you have it. See you in class tomorrow.